Well, that was a truly inspiring talk about Yamanaka. But me standing here, all I'm thinking about how I'm gonna, that's gonna be a very hard act to follow. But before I start, let me start thanking Ian and all the organizers for inviting me to speak at this very wonderful and special occasion of Dolly celebration. As Ian mentioned, we are interested in cellular reprogramming to pluripotency using the Yamanaka factors. But I'm not gonna introduce to you IPS reprogramming following Yamanaka's talk. But what I would say is that cells have been known to be able to change their type from one type to another naturally during embryonic development. But they usually transition from a pluripotent state to a more lineage-restricted state. But unlike this development or programming process, IPS reprogramming, which is go in the opposite direction and is mediated by the 4 transcription factors, is very highly inefficient, and also it's very unpredictable. Only a fraction of a percent of the cells that you start with end up going to pluripotency. So in order to both improve the efficiency and understand the mechanism by which these factors drive cells in the other direction, we're interested how they initiate it to begin with. And why this is important is the pluripotency genes in differentiated cells, such as fibroblasts, are silenced, which means they are within closed chromatin. Closed chromatin contain DNA that is highly packed with nucleosomes, and it's these DNA that these factors need to interact with in order to regulate gene expression. And this is in strikingly different from the state in which pluripotency genes are active in pluripotent state and they are loosely packed and the DNA is accessible for these factors to interact with. So the question is how these factors interact with the genome in fibroblasts in order to activate these genes. And in order to address this question, the first experiment we did is a standard reprogramming experiment as described by Yamanaka, which taken human fibroblasts and infect them with viruses that encode for the 4 transcription factors OSKM, and then wait for 48 hours when the maximum amount of protein is ectopically induced and before transitioning these cells into ES culture conditions, so they haven't received any signal from the ES culture conditions, and did ChIP-seq on all of the factors to map where these factors interact with the genome. So one of the major findings from this experiment was that the binding of these factors, which is highlighted in green signal here from the ChIP-seq signal, lies within DNA's resistant region, which means it's within regions that is closed chromatin, and that was not expected. It's only MYC that binds mainly to open chromatin, but OCT4, SOX2, and KLF4, the majority of their binding sites is within closed chromatin. So the next question, are they guided by some kind of epigenetic mark or histone post-translational modification? So in order to do that, we looked at several histone modifications, and none of them were enriched within these regions, apart from the open chromatin, where it's enriched for the open chromatin histone marks. But the absence of histone mark does not mean the absence of nucleosomes or the absence of histones. As you can see, when we did MNA-seq, which directly map where nucleosomes are, you would see that the majority of these sites are highly enriched for nucleosomes. And these regions include important genes for pluripotency, such as LIN28B, which I will come back to later, that are targeted by these factors. So do these factors interact with the DNA on a nucleosome, on a closed chromatin state? So there are 2 possibilities here. The first one is these factors act as what we call pioneer factors. And this is a select group of transcription factors that can by themselves independently bind nucleosomes, and they precede the binding of other transcription factors. So they prime the genome before the other factors come. While the majority of transcription factors work together in a cooperative fashion and compete with the histones on the nucleosome in order to access their DNA sequences. So to address whether the OSKM factors either follow this way in order to target their genes or this way, what we did was to purify these full-length proteins from both mammalian and bacterial cells, and then reconstitute nucleosomes that we know in cells, as mapped by MNA-seq, contain a nucleosome, and we also know it's targeted by all 4 transcription factors at 48 hours. So this piece of DNA was reconstituted with recombinant histones to make a nucleosome structure. And now we can measure the affinity of all the OSKM factors to the same piece of DNA, both in a naked DNA form and a nucleosomal form. And the conclusion was that, surprisingly, OCT4 and SOX2 were not affected by the presence of a nucleosome on DNA. In fact, SOX2 even prefer binding nucleosomes than naked DNA. KLF4, on the other hand, still binds nucleosome, but with less affinity, while CMYC does not. He prefers naked DNA. So is this also true in cells? To answer that question, what we did, we only looked at the sites where these factors bind on their own, meaning they bind at least 500 base pairs apart from each other, so they won't share any nucleosome, and then measured from MNA-seq data the nucleosome density. And as you can see, you can almost recapitulate what happens in vitro, what's happening in vivo. So OCT4, SOX2, and KLF4 all have the ability to bind nucleosome, while CMYC have very residual activity binding nucleosomes. So why binding nucleosomes is so important? Well, pioneer factors, which are exemplified by FOXA, is a special kind of transcription factors because it has a DNA binding domain that is very similar to linker histone, and meaning that it only needs to occupy a portion of the DNA helix, leaving all this free so it can easily dock on the nucleosome, even when it's wrapped around DNA. While the majority of transcription factors, such as MYC, need to wrap around the whole DNA helix, double helix, in order to recognize their sequence on a nucleosome, so that won't be compatible with the nucleosome state. But none of the OSK factors have this special DNA binding domain. So how do they do it? So in order to address that question, what we looked at the binding motif, which is a special DNA sequence both enriched within nucleosome sites versus nucleome-depleted sites. For OCT4, we found these 2 motifs that are specifically enriched in here, and they don't resemble the canonical motif, whether the nucleosome-depleted sites resemble the canonical motif. And what you can see, this full motif is basically binding half a motif each time. So in order to kind of interpret this data, we looked at the crystal structure of OCT4 DNA binding domain. OCT4 has a bipartite DNA binding domain, has a PO-S and a PO-homeodomain, which is similar to nanog homeodomain that Ian was talking about. When both of these DNA binding domain recognize the DNA, they leave very little space for being compatible with nucleosomes, but they recognize the full motif. But in our data suggests that OCT4 is only using half of the motif by only using one of the DNA binding domain at the time and recognizing half of the site, leaving the majority of the DNA exposed and compatible to bind a nucleosome. So what about SOX2? Similar analysis showed us the only difference between nucleosomal sites and the canonical site is this G nucleotide here. And if you look at the crystal structure of SOX2 DNA binding domain, So the crystal structure of DNA binding domain shows that SOX2 has a high mobility group, which bend DNA almost 90 degrees. And what's surprising is this G that's missing in the nucleosomal motif is right at the angle of the band and is interacting with the tail. If you mutate this tail, which is a residue that interacts specifically with the G, or you mutate the G to another residue, you don't deplete the binding to DNA, but the protein can no longer bend DNA. So what is this telling us? That SOX2, when it's bound to nucleosomes, it doesn't bend DNA because it won't be able to, because there is histones there. But what it's doing is only recognizing this partial motif, which is compatible with the nucleosome structure. And KLF4 is a similar story. The nucleosomal enriched sites is a shorter motif than the nucleosomal depleted sites. And if you look again at the crystal structure, KLF4 has 3 zinc fingers. When all the 3 zinc fingers bound, it'll bind the full motif. When only 2 zinc fingers bound, it leaves more space so it can bind to nucleosomes and only recognize a 6 nucleotide motif. So to cut the long story short, this analysis and others, what showed us what's so special about these factors is their flexibility. So these proteins, their DNA binding domain is flexible and it can adapt to the DNA when it's in a closed chromatin state. Unlike c-Myc, which is more rigid and requires the assistance of these pioneer factors in order to access this closed chromatin and then target the silent genes in somatic cells. But if these proteins are so good at accessing closed chromatin and activating pluripotency genes, why reprogramming is so inefficient? So to answer that question, what we did was to look at the sites that are they not able to bind to. And we noticed these big gaps in our ChIP-seq data where none of the factors are present in 48 hours. But if you look in ES cells, they happily bound at these sites. So we call these regions differentially bound regions. When we looked at their chromatin state by looking at different histone marks, what we found is the heterochromatin histone modification mark, H3K9 trimethylation, which Sir Ian has mentioned in his talk earlier, is specifically enriched at these sites in human fibroblasts, but not in ES cells. So these sites are open in ES cells, and these factors can bound to, but not in human fibroblasts. What's also surprising to see that these large chunks of the genome, which represent the 6th of the genome, are almost identical in a lot of cell types. And some of them are more cell type specific. Also, when we looked at all the ChIP-seq data from about 60 transcription factors, almost all of them are depleted within these DBRs. So this is a more of a general phenomena where histone marks such as H3K9 trimethylation mark a state of the chromatin that preserves cell identity and block access of reprogramming factors such as OSKM to get inside to. So to conclude, what I would like to say by this picture is what we know now is that these OSK factors act like pioneers, and they go to these unexplored chromatin, closed chromatin site and assist each other to access them. But they're not very specific. They don't only target pluripotency genes, and they don't access promoters yet. So first, they need to get into the right promoters, and also they need to overcome H3K9 trimethylation and heterochromatin marks in order to reach pluripotency. And what we're doing in the lab is to use another approach, learn from these factors, and engineer our own factors that are more specific, are binding the right promoters, and they can also easily overcome H3K9 trimethylation and go straight to pluripotency. And with this, I'd like to thank my lab members, and also I've just moved from Kanzaret lab almost a year ago now. I'd like to thank members of his lab that contributed to this project and our collaborators. It's been almost a year since I moved from the US, and I haven't suffered from PAD. And I think this is because this large number of people in our institute who have been wonderful since I moved there and created such a great atmosphere. But I should probably suffer from PAD if I'm looking for any success. Thank you for listening.
Okay, there's a question here.
So the British version of PAD is PED, which is post-Edinburgh depression, and I hope you'll never suffer that. So just a question about this fascinating mechanism. What other biological processes might require pioneer factors that access repressed nucleosome stack structures of DNA, which is another way of saying how specific is this?
Okay. So I'll give you the example of FOXA. So it's obviously involved during early development, but it's also involved in hormone response. So it usually binds to the sites and primes them before it receives any signal. So the signal is very quick. So it goes straight to the genes to the sites that are already primed. I know for data it's been published from Shelley Berger's lab, P53 does the same thing. It goes to sites that are marking or priming the sites prior to their activation. And I don't know how it chooses those sites, but at least this is what they do, and it might be a more general phenomenon.
Some other questions? What about the SOX2 tail mutant? How does it compare to wild-type SOX2 in reprogramming?
So in reprogramming, so the exogenous, we only mutated the exogenous. Yeah. So it reprogrammed the cells fine. But we never mutated the endogenous. I think probably that bending DNA is required during the maintenance. But during the early reprogramming, it can be, yeah, it functions fine.
Marius?
So, beautiful talk. I was curious, so you showed 2 days after infection in fibroblasts, the Yamanaka factors are mostly at the wrong places, right? They are mostly recognized like half sites, whereas obviously at the final product in iPS cells, they must occupy the proper targets. So have you any idea what happens in between? Is there like, do you know, like what are the dynamics when they actually find the proper targets, or are you aware of any other, I mean, in mouse in particular, there's many other reprogramming systems which are way more efficient than in human. Have you any ideas whether, you know, in those high efficiency systems, maybe the targeting works faster or maybe immediately?
So I know exactly from your work that ACL1, for example, is a more specific pioneer factor, and that, in your experience, works faster. But in terms of IPS, so the reprogramming factors, I mean, we have to be very careful because these are the exogenous factor. But the ones that bind and maintain pluripotency, those are the endogenous at the right levels. So the first thing we have to kind of keep in mind is the amount of protein is so far more than in ES cells, especially at the 48 hours. So it can be just a matter of, because we're expressing so much of it, whether the binding all over the genome is required to make the genome, let's say, hyperplastic, so it can respond to the right signals to lead to pluripotency, or whether the actual specific targeting is needed, we don't know. So it can be both ways. So you can argue that, yeah, you need the binding to cover the whole genome to make it more responsive, and then you have to specify it later on, or whether it's so stochastic and only the cells that are hit at the right places will convert. So it can be both ways. But I don't think anybody has been able to say for certain which one is which.
Okay, let's thank Abdenour once again.